and I'll call him back. Okay, so now that I have everyone back, we're going to start talking about cell structure. Really what we're going to be focusing on is organelles. So before we even get into this at all, I want you to remember back to our discussion of prokaryotes versus eukaryotes way back to the first day. Which do you think we're talking about when we're talking about organelles? Talking about eukaryotes, right, when we're talking about organelles, because hopefully you remember our big difference between prokaryotes and eukaryotes is that prokaryotes are really simple. They don't have any membrane-bound organelles, including things like they don't have a nucleus. So all these various structures that we're talking about really going to be seen in eukaryotes, but not prokaryotes, with basically two exceptions. Okay. So in order to understand these organelles, we're also going to be talking about something called protein sorting. So one of the main jobs of your cell as a whole really is to make, package, and then send out various types of proteins. So proteins do all sorts of stuff in your body. We talked a bit about the proteome, and we'll be talking about it even more when we get into our genetics unit. But proteins are really the functional part of our body in a lot of ways. So a lot of the organelles interact with proteins, and a lot of them are involved in their processing. So here you can actually see what a cell, not a standard like little cartoon circle cell, but what a real cell might look like if you traced proteins through them at different times. So here we see a radio-labeled protein in this cell. So they took a picture of this cell at 3 minutes. They took a picture of this cell at 7 minutes, and then they took a picture of the cell at 120 minutes at 2 hours. Okay, so we can see here that our proteins are collecting down in the bottom of the cell in the rough endoplasmic reticulum at 3 minutes. At 7 minutes, they've moved into another one of our organelles, the Golgi. And at 2 hours, we can see they're being released out to the outside of the cell, to the extracellular environment. They're getting there through something called secretory vesicles. Okay. So the first part of this lecture, we're going to be tracing through organelles as they relate to proteins. And then we'll cover our kind of grab bag other organelles that are also important, just not really involved in this process. Okay. So for making a protein, we're going to start in the nucleus. So nucleus is a structure very important that we see in eukaryotes, not in prokaryotes. And it contains the DNA. So it contains our genetic information, and it's encased in a membrane, right? It has a membrane that protects that DNA from anything that happens to be floating around in the cell, but also holds that DNA all together in one space. So we don't have things freely moving, touching the DNA, bouncing away. It's really, really protected inside this membrane. Okay. So the DNA is going to contain the code or the blueprint for our proteins. The next thing we need to know about as we're tracing our proteins are our ribosomes. So they're not technically exactly an organelle. They're not a membrane-bound organelle, but they're critical to this role. So we're going to group them together for now. Ribosomes are one of those two exceptions that I said, things we'd go over today that you would see in prokaryotes. Prokaryotes do have ribosomes. So the ribosomes receive a message from the nucleus. So they're going to receive a signal. They're going to receive the signal in the form of mRNA, which we'll talk about in our next unit. They're going to translate that message into a protein. We'll talk about that process a lot in detail in the future as well. What I want us to know for now is where our ribosomes are. One place we're going to find them is out in the cytosol. So the cytosol, or the cytoplasm, is the watery environment inside the cell. So we've been saying that there's water outside the cell, there's water inside the cell. The cytosol is that water inside the cell, essentially. So some of our ribosomes are going to be free floating just around in that cytosol. And some are going to be bound onto a structure called the rough endoplasmic reticulum. In fact, it's these little circular ribosomes studding the endoplasmic reticulum that give it its rough appearance. Okay. So here we're going to look at the endoplasmic reticulum, right? So we've zoomed in on the center of our cell. Here we see the edge of our nucleus. So this nuclear envelope part, that's our membrane around the nucleus. The red stuff is our DNA. Okay. So we got our nucleus. Right next to it, we have our rough endoplasmic reticulum and our smooth endoplasmic reticulum. So the rough endoplasmic reticulum has these ribosomes embedded in its walls, basically, and these walls are made up of membranes as well. The job of the rough endoplasmic reticulum is going to be continue to make proteins, right? Our ribosomes start to translate code into proteins. So that's a critical thing that's happening here in the rough ER. They're also going to be doing some processing. So they're going to be doing things like protein folding would be happening here in the rough ER. As we're making those polypeptides, they start to make their alpha helices and beta pleated sheets. Then they start to crumple up. We're going to expect to see that happening here in the rough ER. Another cool thing that the rough endoplasmic reticulum can do is make phospholipids. So remember that phospholipids are really the basis for all our membranes. This is where they come from. Now, it's important to tell apart the rough ER from the smooth ER. They look the same, right? They have membranes, they form these kind of tubes, they look kind of like coral or something. But the difference here with the smooth ER is you're going to see no ribosomes. Right, we see these orange little circles on the rough ER. Those are ribosomes. None here on the smooth ER. This means they're not going to be involved in making proteins because you need a ribosome to make a protein. Okay, so they have no ribosomes, so they're not making proteins. But what they are going to be doing is making some carbohydrates, making some steroid hormones, and storing calcium. When in the future, if you talk about muscles, you'll talk a fair bit about the role of calcium. So that will be stored here in the smooth endoplasmic reticulum. Okay. So after we've sent the code from our nucleus to the ribosomes, that ribosome is maybe embedded in the rough endoplasmic reticulum. So as we're making our protein, it starts to fold in the rough endoplasmic reticulum. We're now going to start sending the proteins to other parts of the cell. So in order to do that, we're going to basically put our protein in a little membranous bag, right? We're going to peel off a little bit of the membrane, make a little bubble with it, and our protein is going to be inside of that. This is analogous to what we talked about with bulk transport with endocytosis and exocytosis. So you can see here at the top of this little endoplasmic reticulum, you can see how this bubble is starting to form on the rough ER. And then it pulls away and we have these sort of free bubbles moving out away from the ER. So those are our vesicles. Okay. So it's basically acting like a bag, carrying proteins around the cell. They could carry other things too. Right now we're thinking about proteins. So the place they're going from the rough ER as we're tracing our protein synthesis, protein folding, and then protein delivery is the Golgi. So Golgi apparatus, Golgi body, just plain Golgi, those all mean the same thing. The Golgi kind of looks like pancakes. So they're flattened. And initially they might look to you kind of like the endoplasmic reticulum, right? But what you can see is these stacks are kind of separate, right, from each other. They look subtly different from the ER. We don't have these sort of tubes melding and molding into each other. We've got a sort of smooth stack here. Still made of membranes. So we're going to have our vesicle that's coming from the ER bind into the membrane here on the Golgi. So now the protein is inside the Golgi. Okay. So the Golgi's job is to modify and sort these proteins and also lipids. Lipids would undergo a similar pathway. So it's going to tag them and it's going to kind of plan for the cell where they're going. Okay. So that means at the other side of the Golgi, so we're going to have one side facing the endoplasmic reticulum that's receiving those proteins. It's going to then move through the stacks. The protein's going to move through the stacks. It's going to form another bubble on the opposite side of the Golgi. So here we have a new vesicle. That's going to be sending the protein out where it's going. So in this case, we can see that it's exiting the cell. So this vesicle is binding into our membrane. And then that's spilling the contents outside the cell. So this would be an example of exocytosis. So we say that the Golgi is responsible for protein sorting, processing, and secretion, basically because of this sort of planning and sending aspect it has. Right. So after the Golgi, these proteins are undergoing exocytosis, which is where we have the membrane of this vesicle, this little baggie, bumping into our plasma membrane, that's the outside wall of the cell. And then that bag opens up. Now, as it opens up, the internal contents spill out. Okay. So those are our organelles that we're going to see involved with protein sorting, and that's the basic process. So that's what we're going to be talking about when we look at our problem set today. But we do have some other organelles that we'll want to know about if we want to say that we know something about cell biology. So we're going to go through those now. First of which is the lysosomes. So the lysosomes basically act like the digestive system of the cell. And what I mean by that is if we take something in through bulk transport, through endocytosis, so we have our cell membrane, some little thing bumps into it, and we decide maybe we're a one-celled organism, maybe we want to eat it, right? So we want to eat this, I don't know, bacteria we ran into or something, right? We're going to fold our membrane in. We're then going to peel off that bubble and pull it into the cell. Then we're going to have digestive enzymes in a lysosome binding onto that vesicle. It's going to spill the digestive enzymes into our vesicle as they merge together, which is going to break down whatever's in that vesicle. This means that lysosomes are also good at breaking down other things inside the cell. So in addition to eating food that the cell is picking up in the environment through endocytosis, the lysosomes are also going to be able to do kind of routine cell maintenance. So they're going to be able to break down worn out organelles. Cell can make new organelles, but sometimes, right, we got an old busted mitochondria, maybe we want to break it down, we would use a lysosome. Speaking of mitochondria, so I know it's a meme on the internet, right? The mitochondria is the powerhouse of the cell. The reason that is true is that they are making ATP, which is our cellular energy. So they are making this energy that's going to be able to fuel basically all of our cellular processes. Now they're able to do that because they have a specific structure, and we are going to get into metabolism and exactly how ATP is made at the end of this course. But for now, I want to take a just basic look at what a mitochondria looks like so we can identify it. So our mitochondrion looks something like a kidney bean from the outside in shape, has this outer membrane. The cool thing about the mitochondria is it then has another inner foldy membrane, which means that we have kind of a space between the outer membrane and the inner membrane. It also means that we're going to have a very center sort of region that's protected by two membranes, right? So we're going to see as we go through metabolism at the end of the course, how different molecules move into the center of the membrane, then out into the space between the two membranes, then back. And that's going to be very critical to how making ATP works. But for right now, just know two membranes, a space between them, and a space on the very center. Okay. Mitochondria are also kind of cool and weird. There are some pretty wild ideas about why eukaryotes have mitochondria. The basic idea is that some very, very ancient cell was cruising along doing its own thing. It was pretty big. It ate another cell. By this way, we do endocytosis, right? Sometimes single-celled organisms do this for food. It ate this other cell, but this other cell happened to be super good at producing energy, and it started to leak into the bigger cell. So instead of digesting it with a lysosome, it formed this sort of symbiotic relationship with this other cell. So because of this, mitochondria actually have kind of unique properties. They have their own DNA. They can replicate and grow on their own using their own DNA, which isn't true of other organelles. It also means you can trace mitochondrial DNA. Mitochondria are specifically inherited from your mother. So you can trace female lines back. They're pretty neat. There is another organelle that scientists theorize started out the same way the mitochondria did, kind of. It's found in plants. So chloroplasts are a plant organelle. Their primary role is to do photosynthesis, which is where you make sugars and oxygen from light. Then the sugars can then be used for energy. They're pretty cool. They're also what we call semi-autonomous organelles. So our two semi-autonomous organelles would be mitochondria and chloroplasts. Chloroplasts are only in plants. So they can also replicate on their own, grow on their own. They have their own DNA. Really weird, really neat. The final thing we want to talk about is the cytoskeleton. So the cytoskeleton is not made up of membranes. So you would expect to see this broadly in lots and lots of types of cells. The cytoskeleton is itself made of proteins. So the cytoskeleton is kind of like a scaffold. Like if you've ever seen them doing repair on the front of a building or building a building, right? You know how they put up a scaffold in order to kind of hold everything in place while they're making it. The cytoskeleton looks like that, but inside your cell. So it's always there, but usually it's not drawn in, because if it was, it would look kind of like this image on the right, make everything super messy. So just to kind of orient you, this yellow thing with orange inside, that's a picture of a mitochondrion. Over here in pink with darker pink little dots on it, that's some endoplasmic reticulum. You can see we now have this kind of webbing kind of everywhere. So that's the cytoskeleton. It's made of proteins. While we don't always draw it on, it is super important because it's what allows cells to have their shape. It holds the organelles in place. And some of those organelles, it's important that they're near each other, right? Like the endoplasmic reticulum has to be kind of facing the Golgi in order to send the proteins, for example. It also allows for things to move through the cytoplasm. And specifically, it kind of allows the vesicles to travel along the cytoskeleton like little cars. And when the cytoskeleton changes shape, so if those proteins move, right, if we had a scaffold like this, and then suddenly they move like this, can actually cause movement of the whole cell. We'll see that there are analogous components of the cytoskeleton outside the cell as well. Those are a little different, but we'll see some related proteins outside the cell. Finally, because this is a basic rapid fire cell biology course, you should know that plant cells are different than animal cells. We're mainly thinking about animal cells. We're pretty biased, right? So those circular cells that we're drawing with the nucleus in the center, the membrane, the endoplasmic reticulum, or mitochondria, or Golgi, we're usually drawing an animal cell. But a plant cell is going to have some more specialized organelles. So I think they actually, they don't click on. So just in case you see a plant cell, right? In addition to having an external membrane, they would have a cell wall, so a rigid cell wall. And we talked about how that is partially made of carbohydrates, right? They're going to have chloroplasts. So they have both mitochondria and chloroplasts, while in the animal cell, we only had the mitochondria. And sometimes we'll see organelles that hold like colors. They have a lot of cool things, but we're not going to focus on them for the purposes of this course. Oh, vacuole. We did mention the vacuole before, but just kind of to emphasize here, when we were talking about osmosis, we talked about how if the cell is in a hypotonic environment, so we have a bunch of water moving into a plant cell, that cell gets rigid, basically because this vacuole swells and puts pressure, outward pressure on those cell walls, making our plant nice and crispy, basically. Okay, so now I'm going to pull up our problem set and just kind of orient us to that. Not on that. Got to open the right window. Okay, so the problem set we're doing today is on protein sorting. It's pretty neat. It's actually on protein sorting with a focus on looking at a kidney disease that causes a type of diabetes. So these first couple questions are actually orienting you to what a kidney is and how it relates to our concepts of transport so far. So for this first question, basically, if you don't know what a kidney does, just kind of Google what a kidney does. I trust that you can figure that out. So then these next two questions, what we're asking you to do for these next couple is to read this diagram. So this is showing you some of the pathways you would see inside a kidney, and different regions will see the membranes of the cells are permeable to different molecules. So we'll see that different things can cross membranes in different regions. So we're asking you to go through and look at specific points here in our little kidney diagram to see what can go across the membrane. So that's in boxes. So you're really just doing a guided reading of this diagram for number 2. Okay. So then for number 3, what you're going to do is draw a little cell. We've done the initial drawing for you, and you're going to add in some of the organelles. So you're going to be adding in organelles you need for protein sorting. So go through the slides, take a look at which ones those are and where you would want to position them in this cell, label them. And then in number 4, we're going to start thinking about this disease, NDI, which is a type of diabetes, not one of the common ones. It's kind of different, but it's a water balance disease, actually. And we're going to see that that's basically due to a miscoded protein. So sometimes this protein gets messed up. It's a protein that's partially responsible for our water balance. So you're going to read this little paragraph. You're going to critically think about it. You're going to then look back at your cell, put a star where that protein should be, okay? Should be in its normal function. And then you're going to go back through the organelles you've drawn. You're going to think about, if we're making this protein wrong, if we have this disease, where is this going wrong? And then finally, what effect overall would this have? So in order to do this final point, this what overall effect would it have, look back at this normal function, basically, of the protein, so what that protein's supposed to be doing, and think about what would happen to the cell and to the cell's function if it couldn't do that process. And I'll be in to help as you guys need help. But I know people get a little bit shell-shocked seeing this diagram initially, so I thought I'd walk you through it. Okay. I'm going to send us back to our breakout rooms to work on this. So take a couple minutes to look at it together as a group, and then call me in to help. please.